So, okay, the technology seems to work. I better get going. The title for that is Sustainable Crop Production Under Increasing CO2, but basically the content will be, I would like to introduce you to some of the more recent highlights maybe from our research program, which we call the AGFACE, the Australian Grains-Free Air CO2 Enrichment Program. The background of all our work, as this is in crop production and crop science, is of course, I would say, the growing global population and the growing demand for food. And this is a projection taken from the FAO, the Food and Agriculture Organization of the United Nations, and extrapolated to say around 2050. And according to current trends, we would have to increase food production very significantly. You see the blue line is just a continuing trend, but actually the demands are even above trend. Those would be the red lines, and to keep our increasing global population fed, food production needs to be about doubled until 2050 over, say, 2000 values, which is an enormous task, given that the historic increase in global, and that's only for cereal crops, but it's basically the same for all food. But the historic increase, the past production increase, was mainly achieved by actually taking more land into production, by irrigating more land, by putting out enormous amounts of fertilizer and so on. And it was not a lot achieved by actually increasing the yield per area. So we are running out of potential croplands, and of course we don't want to deforest more of the world for further agricultural production. So at this stage, optimists think that the way to achieve that is by sustainable intensification of food production. Now that term has been coined by Tim Reeves, who is a professorial fellow at this university, and at the time he was head of SUMMIT, which is the International Maize and Wheat Breeding Center, mainly for third world countries in Mexico. And this is a definition about sustainable intensification, what sustainable means there, but what intensification means that we should produce more crops, more food on the same area using less resources than we do now. And that's, of course, an enormous task, and especially in the last decade, the historic development in the last decade doesn't really make us more optimistic, because the historic increase rate has not been sustained globally. So that's sort of the global background, and of course all this comes in the face of climate change. Now this is taken from a few years back, the GARO Climate Change Review for Australia, using different climate predictions, but basically you see either stabilizing at 450 ppm the CO2, and then the extrapolated climate from there. Now that's already way too optimistic. We are tracking way higher than that, and our globally, in the moment, our projections would be we run, I'll show you the CO2 trend in a second, but we would run by 2100 at about 700, maybe even more ppm CO2 in the atmosphere. So probably the trends are much worse than this model change, but even this model change, you see by 2030, there may be still some increases in crop production in some areas, but by 2100, certainly none of the projections predicts any increase in production in Australia, but only decreases. So we're dealing with enormous increased demand globally, and with the adverse effect of climate change. Now, climate change, of course, is directly caused by increasing CO2 in the atmosphere. And without even going into the discussion what causes the increase of CO2 in the atmosphere, it's clear that it's happening. And this is a measurement series done by the Ocean and Atmosphere Monitoring Systems, interestingly run by the Department of Commerce in the US. But they have nice animations on their website, and I pulled that off there. And now let's see if it even works. What it shows to the left is over the latitude, the average CO2 concentration per month in the atmosphere. You see in the northern hemisphere annual fluctuations that are quite strong, not so much of that in the southern hemisphere. The red dot in the middle is the Mauna Loa station, quite famous on Hawaii, which has the longest running, let's say, reliable atmospheric CO2 measurement series. And the blue station is one on Antarctica on the South Pole, has also long series. There is one station here, which is Cape Grim in Tasmania. It's quite famous because it's supposed to be the cleanest air measurement station globally. But you see clearly this was now from starting somewhere in 1980 to 1992, and the increasing trend from 320 to 350 is pretty clear. Here is the time trend. I'll probably have to do that here. So here's the trend over the years, and you see these fluctuations that you can see on Mauna Loa. The map shows all the stations included to here. Let's do a fast forward now from 1990 to 2010, and we see here touching the 400 ppm. That's a nice threshold, easy to remember, 400 parts per million CO2 in the atmosphere. We crossed that globally 1.5 years ago, and we just crossed it also on the southern hemisphere, where values are generally a little bit lower because the atmosphere is still a little bit cleaner. There are less cities and less population, but we crossed 400 ppm at Cape Grim in Tasmania about a month ago, I think. So let's go another fast forward. This actually stops here. But there is an extrapolation now backwards, and the green series are still probably less reliable direct measurement series. And then if we go further back, of course, to pre-industrial times, there were no direct CO2 measurements in the atmosphere done, but the extrapolation that you see here is from ice core data where fossil atmosphere bubbles are analyzed. And we are very confident that pre-industrial CO2 was about 278 ppm, 280 ppm. So you see that's actually a big change. And it was for a long time stable at that level. So basically, plants have evolved for that, and then we have a very fast change just in the last 100 years. CO2, of course, is the main plant nutrient. So CO2 in itself will have an effect on all plants, on all ecosystems. And this is just a citation by Louis Siska, who worked a lot in elevated CO2. And despite all kinds of research done, there is a bit of a neglect that CO2 in itself has a strong effect as well, not only the knock-on effects, of course, of climate change. And in a way, it's the hardest one to study. It's the most we know that's happening, but there is no surrogate for studying future conditions. For higher temperatures, for example, for most areas, you can go into warmer areas and then have a good idea of what would happen if it gets warmer in that place. Or if it gets drier, you can move across the rainfall gradient to areas that are now drier and have a bit of an idea what might happen. But CO2 is the same more or less globally, so it's much harder to study. And that's where our experiment comes in. I want to introduce you to the AGFACE, the Australian Grains-Free Air Carbon Dioxide Enrichment Program. Now, this is our field site out in Horsham, in the Wimmera in Western Victoria. What you see on that, maybe just as an introduction, in the background, the white tower is the CO2 tank. And this a free air carbon dioxide enrichment installation, quite generally called FACE installation, enables us to blow CO2 onto an ecosystem, a crop, even on forests, as the colleagues in Western Sydney University prove, to study the effect of elevated CO2 of the future atmosphere on the ecosystem without any artifacts, enclosure effects, glasshouse effects or so. So that's a phase system. So CO2 from this tank is delivered to the plots. In the foreground, you see one of our plots. All these red labels are all different small experiments going on. I come to the technology of the plot in a second, but you see in the middle of each plot is an anemometer, a wind measurement device that controls the CO2 delivery. Okay, just to introduce the program here and get that out of the way. Of course, what I show here is mostly not my work, but the work of our team. So the University of Melbourne AGFACE team is actually very small, I would say. So it's Maurice and Marcus as the scientists, and Sabine as the other chief investigator, and Sam has recently left us, so our team is even smaller. The AGFACE is a collaboration with the Victorian government, now called the Department of Economic Development, Jobs, Transport and Resources. I'm very proud that I didn't stumble on that one, but they adopted now the easier, their research division now adopted the easier name Agriculture Victoria, which I was told was used about 25 years ago as well. So, and the team at the Victorian government is named here. There are many more senior scientists in that area than on our side. And I hope you all, well, that the ones local here probably know all the research students attached to that program. Okay, now the one thing with studying CO2 is that it involves such phase technology, which is quite high level, and it also involves quite a lot of CO2 use, and that's relatively expensive. And so you need relatively large amounts of core funding to run such a program. And of course, we are very grateful to our main funding bodies, that is the GRDC, the Grains Research and Development Corporation. So that shows why it's called Grains Phase Experiment and why we're doing mainly grain crops. We also got very significant funding by the Australian Government Department of Agriculture and Water Resources, as it's now called, and some support by the Australian Research Council through different programs. If you want to see more, there is the web page with some information. First, putting it in context to globally and within Australia, the one thing that is very important about AGFACE, about the site there, is that it's in the middle of the Australian grain production belt. The Australian wheat belt is in black here. That's actually the map from the FAO. And Horsham is in a region called Wimmera, which is the main grain production region in Victoria, together with the Mallee, which is northwest of the Wimmera, also in that area. It's also a relatively low rainfall area for grain production globally, and for the last, I don't know when they shut it down, but there is no irrigation agriculture in the Wimmera anymore because there was never water in the Wimmera River for a long time anyway. Globally, and this of course is a very odd looking slide, and it's actually the back of a t-shirt that we got on a so-called World Crop Face meeting organized by the colleagues in Tsukuba in Japan. And there are a number of similar comparable face experiments around the world, all operating in different agricultural production systems. So the longest running ones are, well, ours is actually getting there as well, but the longest running ones are in Illinois in North America. We may call it the soy face because soybean is the main crop they study there. And actually our colleagues in New Zealand run a phase experiment on pastures, including sheep on the phase rings and so on. And that was running also for a very long time already. And then the colleagues in China and mainly in Japan are investigating rice. And in Europe, there are a number of phase experiments off and on, depending on funding. And they're studying different kinds of crops, often crop rotations and so on. So AGFACE is in a network of global phase experiments. And the special thing about AGFACE, I would say, is that it's at the very low end of productivity and of rainfall. You see here the relative or the grain yield of different phase experiments where they have grown wheat one time or the other in relation to the annual rainfall. And you see that the AGFACE results are at the very lowest end compared to all the other phase experiments. Probably the first one and one of the famous ones was run in Maricopa in Arizona in the middle of the desert, and you would think, well, actually, clearly that is lower rainfall, but it is a continuously irrigated production system. And you see the Maricopa data have actually fairly high grain yield in tons per hectare. I don't know if you remember the number in the one slide about global cereal production, but the average grain yield of wheat, for example, globally is just above 2 tons per hectare. So our phase system is actually very representative for a large part of global wheat productions. And so that's where its global importance comes in. Now, a few pictures and explaining how that works. This picture was taken by our senior technician from the Victorian government, Russell, from an airplane, and it shows our field setup. The phase plots are these ones, and they are embedded in a background cropping system so as to minimize all kinds of canopy effects. You see some of them are light and others are dark. That's because in that year we had a crop rotation growing, so half of the plots were grown with field peas and the other half with wheat. So wheat is the dark one here because the background crop is, I think it was pea or fava bean or some legume as well. So how does that work? This is now one of these plots, and you see in the center of the plots, as I showed before, is this wind speed and direction sensor and a central CO2 sensor. That's where the CO2 target value is measured. And we actually target 550 ppm, which according to current predictions is about 2050 values. But we're actually accelerating the trend every year, so we may already be earlier at what we investigate now. You have these tubes, we call it a ring, but it's actually an octagon of stainless steel tubes around the plots. And these tubes have, as you see in the little picture here, little holes drilled in them, and they're pointing to the outside of the ring, the outside of the plot, and depending on wind speed and wind direction, CO2 is delivered into the oncoming wind. And then it's mixed by the wind blown over the plot, and the target in the middle regulates the CO2 delivery to stabilize it at 550 ppm. It's quite surprising how stable that is and how well it works, because generally Mahabubur Bola is the research engineer actually running the technology, and he did quite a bit of investigations on the stability of the delivery and so on. And the variability, once you're sort of half a meter away from those tubes, is actually pretty good. It's generally better than any glasshouse or chamber system. Also, that's maybe harder to believe, but it's because glasshouse turnover takes normally much longer. So this is our field setup. I might go a little back now to CO2 effect. CO2 in itself is, of course, the main plant nutrient. So there is a thing called CO2 fertilisation effect. With higher CO2, we expect plants to grow better. And that's known for ages, actually. It's even used in horticulture. These are horticultural glasshouses from up in Queensland, and they are supplied with CO2 to keep an atmosphere of 600 ppm or so in there, especially for tomatoes. That seems to be very, very good. There have also been lots of scientific studies on elevated CO2 since about the 90s. So we know in general a lot about the effects, but I will show you what the specialties now are. Oh, that was too fast. So the physiological explanation is easy. Don't worry too much about the details. This is a textbook CO2 response curve for photosynthesis. So photosynthesis is, of course, the determining plant reaction, that the biochemistry that determines plant life, and it's directly dependent on photosynthesis. In this, the fat black line is the overall response curve. So CO2 is on the x-axis here, called CI, because it's internal CO2, and A means assimilation rate. CO2 assimilated into the plants per square meter leaf area and second. And so if you go up, in the moment we're sitting at 400, if you go up to 550, you can expect assimilation rates to increase. That's true for so-called, just as an aside, that's true for so-called C3 plants. There is a different type of photosynthesis, so-called C4 plants, which are already saturated at nowadays CO2 rates and won't increase their CO2 fixation just because they have more CO2. That's an interesting aside for one reason, because there is lots of research efforts going into trying to make rice a so-called C4 plant with all kinds of biochem technology. So if that takes more than 20 years, then it's probably totally obsolete anyway, because those plants will not have an advantage, and the normal so-called C3 plants may catch up by then. Okay, maybe another curiosity. Phase systems are not as new as we may think they are. So the first ones were installed at the end of the 80s in the US, but this is actually a paper describing what I would say the first phase system in 1927. And at the time, of course, it was meant to be a CO2 fertilization system. And I recalculated the CO2 delivery they had, although the measurements were done with wet chemistry and so on, so quite difficult. But they reached about the same concentrations that we had, or they were at least targeting about the same concentrations. So that's just an aside that we're maybe not as novel as we think we are. Talking about the CO2 fertilization effect, yes, we see that also in our system. Plants generally grow better under elevated CO2. And you see to the left, the plant is from elevated CO2, a wheat plant, and to the right from ambient. And that's a quite typical result over all these years. We were running different experiments now. Just a comparison between global results overall and ours in our drier and lower production system. And actually, they line up pretty well in the overall numbers. So, for example, about 20% increase in biomass by increasing CO2 from 400 to 550. Maybe just as a step back, think about that. 400 to 550 is actually a big change. It's about, what's my calculation now? I don't know, I can't do it quickly, but nearly 40% change in the main nutrient. So one nutrient changes by a lot. If you had, for example, 40% change in water supply or rainfall, clearly that would be a major concern for all farmers. If you, for example, eat 40% more carbohydrates, but leave everything else the same, it would also have major concerns for your nutrition. And that's one of the things plants have to grapple with as well, because there is some sort of imbalance developing. So the first 2 points, biomass and yield, are stimulated by CO2 because you have more growth. The main nutrient makes plants grow a bit better. But then the second 2 points, for example, protein, not only in grains, but in all plant tissues, we found that that decreases. The protein concentrations in plants decreases because protein is mainly dependent on nitrogen. the second most important nutrient for plant life. And nitrogen is already limiting for agriculture, so maybe there is a limitation. I will come back to that. And many other nutrients, many of them very important for human nutrition, such as iron and zinc, are also decreasing. In our case, in grains, because that's mainly for grains, but that's true for basically all plants, and also if you eat other plant parts. So our current research themes are around these changes in crop nutrition. I come to the other resource, water, in a minute. On the one hand, and that was mainly supported by the Commonwealth Department, Ag and Water Resources, and then how to maintain and increase and use the CO2 fertilization effect for wheat yield, and at the same time try to maintain this grain protein and other yield quality components. That's our second big research area. And I just throw out now some highlights in these research areas without going too much in depth and without stealing the thunder of all our research students and individual researchers if they give a presentation over the next month or so, years or so. Okay, so resource use under elevated CO2 was not seen as a problem in agricultural system for a long time in all these experiments, because we thought, well, we manipulate resources anyway. We put on fertilizers, we often put on water, so any changes there may be a problem in natural systems, but not so much in agronomic systems. And that has clearly proven wrong. So one of the big topics we have is looking at crop resource use under elevated CO2. Maybe why that has been neglected was that, as you've seen in the comparison of FACE experiments, most others were in high yielding areas, high rainfall, mostly also continuously well-watered or continuously rainfall supplied areas. Now we are in a very different situation where crops, and this is from the GAC, the information about cereal crop growth stages. Our crops grow over the winter into spring, like sowing is about now, and harvest is in early December, so just in early summer. And anthesis is flowering, or anthesis in cereals is the key physiological state. Until then, plants are very physiologically active. But in our environment, after that, they run into a phase that's called terminal drought. So it's the seasonal drought in a typical Mediterranean environment. It's quite strong here, and basically from flowering to then producing the grains, the so-called grain filling phase, that can be a very critical phase. It can depend a lot on how quickly that dry down happens, how much, even if it's little, but how much additional water is available here in these phases. I keep forgetting that I shouldn't show it there. So any water supply here might be very important. And that's something most other phase experiments have missed because they don't have that extreme terminal drought phase. It is, however, very typical for many crop production areas around the world, and arguably for the more critical ones that are often in the poorer countries and large parts of the population are dependent on such systems. So in such I just give you one example about water use. In such a system, let's go back on that, water use or water use efficiency can be very important, because if the crop, as I said, if the crop has more water available here during these phases, just a little bit more, it may make a big difference for how long they can keep on photosynthesizing and filling grains. Now, in Australia, led by CSIRO and ANU mainly, there was a lot of emphasis put on water use efficiency of crops and breeding crops to be more water use efficient. And the idea is water use efficiency means how much water or how much yield is produced per liter of water, as an easy way of putting it. So if water use efficiency is greater, then the crop uses less water during that phase here, and because it lives on soil water resources for the rest of the season, it may still have more of those. So we were looking at that, how that changes, because elevated CO2 is known to change water use efficiency. And I mentioned that in a minute when I show the results. So this is actually from Sabina's work on water use efficient varieties of wheat as they have been produced and are now commercially available. But one of the main things here, which I haven't explained before, is that elevated CO2, which are the red lines here, generally increases water use efficiency of plants. Why? Well, the one thing is water use efficiency is production per liter. Production comes from assimilation, and assimilation is greater under higher CO2. But the other interesting physiological aspect of CO2 on plants is that plants also close their stomata a bit more. You know, stomata are the pores that plants have to open to let CO2 in, but they also try to keep them closed to conserve water. So plants sense that if they have more CO2, they can afford to keep the stomata more closed. So it's a very typical reaction that plants have lower stomatal conductance under elevated CO2. If you put both together, the effect on water use efficiency, which is assimilation by stomatal conductance divided by stomatal conductance, is even greater. And then for this purpose, I think we don't go too much about the differences between cultivars, but what we showed here is that water use efficient cultivar, we can pick up the differences to a Czech cultivar. It does what it is advertised to do. It is more water use efficient during some times of the day, if you look here. But the interesting thing may be that this is actually a positive effect under elevated CO2, and we were not sure about that because elevated CO2 increases all water use efficiency. You see, I go back to show you, the red lines are actually much greater than the black lines. So you would think that little difference between red and black line, which are the cultivars, may not make much of a difference anymore when you have so much better water use efficiency. So this is from our colleagues in CSIRO, where they developed this cultivar, and they always plotted the yield advantage of the cultivar here on the y-axis compared to the yield of all cultivars in that area, which they call the environmental yield, or you can also use a Czech cultivar that doesn't carry this high water use efficiency trait. And they find in low yielding years or environments, over here on the left, maybe a 10% advantage for their cultivar, and that's way enough for breeders to sell that. Now, if we compare our AGFACE data here, we find that same effect actually only under elevated CO2. Now, we were told by the scientists who developed that they're not surprised that in our environment that cultivar didn't show much of an advantage because we supposedly still have a little bit more later rainfall than they had where they developed it further up in New South Wales. However, under elevated CO2, it appears that our environment becomes much more like the environments they developed this cultivar in. And we get about the same relationship between the yield of the Czech cultivar and the yield advantage that they had in their collective of field data under ambient CO2, but further up in New South Wales and Queensland, where the crops grow nearly only on stored moisture. So anyway, this is one, we're looking at many different traits that breeders are currently trying to get on the market, and we evaluate them under elevated CO2. So in this case, the result is quite nice because this superior transpiration efficiency trait may become even more important, or at least important in areas where it's currently not that beneficial. This is just to show some current data that Alireza produced in his PhD. These are, he worked out a stomatal conductance model using published ways of modeling stomatal conductance, and you see here all his measured data against the model data, but this is under ambient CO2 and under elevated CO2. What I want to show here is only, this is a bit of an artifact on the way the model works, but you see generally that under elevated CO2, stomata don't open as far as under ambient CO2. So the stomatal conductance, the maximum stomatal conductance, maybe look in the X direction, that's at the measured values, is quite a bit lower than under ambient CO2. Now, this is all in a way good. So you have higher water use efficiency, you can check on whether these traits still work. Does that not mean that we have water savings? Because we have higher water use efficiency and the stomata close one. Actually, that was the going opinion for a very long time, because it has been measured in current highly watered systems, especially the soy phase in North America, which is in a very high rainfall zone on very deep, the best soils probably from around the world. And they found that canopy transpiration, means the water use of the crop directly, is always less under elevated CO2 and production is more. So it really looks very nice for our future. At least some of the bad effects of climate change may be offset by the direct effects of CO2. There is a very interesting, let's start this way. We tried to evaluate that in our experiment, and we didn't have at the time a technology that the colleagues in the US had. So our results were a bit inconclusive. We seemed to find that in some years, the overall water use of the crop was actually more, and in others it was less. So that's clearly a question, and we were not sure about our data, but there is quite a nice recent paper out. You see the citation there if you're interested. It comes from a totally different angle. What they did is they observed Australian water catchments historically, the last 30 years or so, and said, well, CO2 was creeping up during all that time. Can we already see a difference? And they used a remote sensed vegetation index, the so-called NDVI, which is a vegetation greenness index, to evaluate large-scale changes in vegetation in these catchments. And the one thing they found is you can already see the CO2 effect on vegetation. So vegetation is thickening in many areas in Australia, and even after you correct for all the rainfall variation and climate variation, you have a very strong thickening effect with increasing CO2. So that was one of their conclusions. But at the same time, they measured direct water yield from all these catchments. And from water yield and the area of the catchments, they could estimate the water balance with all the rainfall data they had. And the interesting thing there is, remember, the going opinion was that under elevated CO2, vegetation will become more water use efficient, will save more water. However, these are just their summary results. You see the mean CO2 sensitivity for all their catchments, their ecosystems, goes in the other direction. It goes positively for ET, which is evapotranspiration of the vegetation. It also goes positively for NDVI, which is the greenness index. But that depends a bit on what ecosystem you're looking at. So the very wet ones seems to be very messy, which are mostly, these would be the ecosystems where we have experimental data from phase experiments, quite wet, normally temperate ecosystem from somewhere in North America or Europe or some tropical ones. But the ones that cover most of Australia's area, well, not most, but the most important catchments for Australia or any Mediterranean area are actually the ones that are not that wet and not that dry. And in those catchments, they found that actually under elevated CO2, water yield from the catchment was getting lower and lower, even after you took out all the climate effects directly, rainfall effects and so on. In fact, evapotranspiration of the vegetation actually increased. Now that seems a bit counterintuitive, but it depends on the balance between changes in water use efficiency and actual changes in biomass. If you have much bigger plants, these plants may be more efficient per biomass in using water, but it may be so much more biomass that overall you have higher water use, which seems to be the case for most of Australia, which is really concerning because we are already in a water scarce environment. And you see, this is then runoff, because that's what they were interested in the first place, water for our use. And it decreased very significantly for those mid-range catchments they investigated. So whilst this is an observational study, using correlations over the years, I think it raises a very important point. So we put in now lots of experimental measurement systems to look a bit more detailed on crop water use. And this is just, I pulled it out from quite raw data from Shihab and Marcus, soil moisture profiles under the crop. And this is just one particular depth. I think I took out 30 centimeters, but it's more or less the same in some of the depth. And we're talking about, does elevated CO2 now save water under the crop, or does it use more water? And the original idea was it would save water because crops would use less water, so they have more water available later in the season, so you would end up with higher soil moisture later in the season. You see the season progression here. Now, you know the blue points, we had well-watered plots in these rings that are the green ones, and we had just rain-fed in the very dry season plots, these are the red ones. And then you have the dotted lines, and the triangles are elevated CO2, and the full lines are ambient CO2. So, I think the interesting thing here is, this is changes after a rainfall event. So we had a quite significant rainfall event within the season. And we don't see much of a change here, although you see in the well-watered, it's even the other way. There's no sign of water saving and elevated CO2 under these conditions. I might say that in the previous season, we thought we saw the predicted water savings. So it really depends on the season, and that may explain why it depends on the season. There was a rainfall event here, and then you have recharge of water into the soil. And that seems to depend a lot on whether the crop was grown on elevated CO2 or not, and when that rainfall event actually happens. So there wasn't much on the already well-watered plots, which is not surprising because you can't saturate the soil more than it's already nearly saturated. But for example, you had a lot of water recharging under the ambient CO2 crop and not much on the elevated CO2. So that may have to do with the biomass that's there and with root structures. Maybe CO2 makes plant roots grow differently in different strata, and so they may capture the water quickly and don't let it into the soil. So that was quite surprising, and of course we're looking now at roots as well. This is the so-called Minirhizotron or a root scanner. And you see the first view of root data. This is actually not on wheat, but on lentils. And these, the two columns of graphs are two different lentil varieties, and the dashed lines are elevated CO2, and the solid lines are ambient CO2. And you have different depth in the soil. All we can see here, or we can see a lot, but the main thing is that actually the effect of CO2 on below ground may well depend, first of all, on the crop variety, and especially on where you look, how deep in the ground you look, and then also when you look. Because you have, for example, if you looked here, you say, well, CO2 increases root growth, well, generally. If you look a bit deeper, you don't see that, although that's where as many roots are as at the top. If you look a bit later, for example, and in this cultivar, you would conclude, oh, elevated CO2 is actually decreasing root growth. And that's makes a very complicated picture, because of course you have an interaction between where the water is, where the roots are, and how it can be used and how it can recharge soil moisture. So this is our current ongoing research, and I think that's quite exciting. We do something else. We have another experiment there which we call the soil face. It's called soil face because we have these soil cores taken from different places and exposed under face conditions. So we can also see whether there are differences depending on the soil conditions, because otherwise the limitation is you're always sitting on one plot. And the good thing with this is we can put lysimeter balances under these soil cores and actually measure directly the water used by the change in weight. So that's another ongoing project where we hope to learn more about the timing of water use in dependence of elevated CO2 or not, and in dependence on the soil conditions. Okay, we have another big area, and I give you only a couple of highlights there, and that's the change in crop nutrition and grain quality. So resource use is of course not only water but also nutrient use, and mainly nitrogen. Without giving many results, because that's mainly Sabine's research area and many of her students, and they will present probably very detailed results in their own talks, but just a background to that as well. It is actually of concern that plants under elevated CO2 have decreased protein and zinc and iron levels. These are the main three concerns globally for nutrition. These are data from a summary paper done by a guy who is actually a human nutritionist and including our data. And you see that there is a 5 to 10% change in cereal protein, iron, zinc under elevated CO2. And he extrapolated it also on what it means for global nutrition, and it's actually very significant, especially in poorer countries where people really depend on grains for these parts of their. You wouldn't depend on iron and zinc and grains. If you buy a cereal, it's chemically fortified with iron anyway. But of course, many countries can't afford that, and they would be directly dependent. So there is a concern. In Australia, it's also a concern that these traits in the grain actually determine bread quality. So our colleagues at the Bread Quality Lab in Horsham, Joe Panozzo and Cassandra Walker, they're doing baking tests with the grain we produce. And actually, consistently, we see that the future CO2 grown grains don't produce such nice breads. And so actually, at the first time, we also thought, well, you know, bakers just add more soy protein. But there was a recent article about that globally. I think it was maybe Canadian bakers. They were already complaining that they have to invest so much in buying additional soy protein because the grain quality is so poor these days. And I think there is quite some relationship already to the CO2 effect. Maybe just a very quick thing. The original idea was, okay, plants grow bigger, so they need more nitrogen. So maybe they can't get enough nitrogen, and therefore they can't make that much protein. Very simple. It's a so-called biomass dilution effect or something. Now, we were in a way lucky because for the low production that we had in our site, we had fairly high nitrogen in the soil because the production was so low. So we could play with giving them really super optimal nitrogen, and that should fix it, shouldn't it? And actually, we did that over a number of years and many environments. And in the end, we saw that even if you put on more nitrogen, which is not desirable for many other reasons anyway, you don't fix the grain protein problem. So it's not purely this. And our thinking in the moment is that there are a number of different options, especially in our environment, from flowering to maturity. There is a lot of internal redistribution of nitrogen going on, and apparently CO2 changes all these processes. Because there's more carbohydrate, you get these interesting imbalances in all the biochemical processes. I don't go into more detail here because it's ongoing research, but I think we have already quite a few nice and exciting results that can be used for either for the breeders to know what to look at, and breeding for nutrient use efficiency and grain protein is another apparently very valuable thing. But also for nitrogen management. How can we put on fertilizer in an optimal way, and how do we need to change this practice as CO2 changes? Okay, so maybe just a few points that we're currently following. I think the overall water use is certainly an interesting one in our ecosystems, especially the timing and spatial distribution of water supply, water use of root space, and there are clearly already interactions with nutrients as well, because the roots can only take up nutrients from the soil where they actually can go. Okay, I think I'll leave it here. It's open to questions, of course, and you can also question all our team members on the more detailed results they're working on in the moment, I guess.
Well, thank you, Michael. Technology still, yes, okay, so technology as expected with Michael is still not 100%. So the only microphone that's picking up anything is from the computer here, so this won't work. So I might hand over to Michael, because if you have a question,
okay,
he can paraphrase it so that the listeners online are able to hear it. Questions? Put you off now, haven't I? Yes, Nina.
Yeah, I have a question. With the other phase studies around the world, have they also observed the same, the less protein in the core?
Yes. Yep. Okay, I showed that in one of the things where our results were compared to the global results, and it's the same everywhere. And that figure from the Myers from our Nature paper, that actually compiles lots of data from many different phase experiments.
So the question was about whether those nitrogen nutrient sort of protein results were similar across other phase sites.
Okay, next question I remember to repeat, sorry.
So, okay, I asked Mike, in the same Nature paper, so effect of elevated CO2 was quite pronounced at cereals and legumes, but not so much on peas and other. Like there was a big arrow bars, which means so if you stick to peas and all those other vegetables, they're safer.
Okay, the question was that in the graph from that Nature paper, the effect of elevated CO2 on decreasing grain protein was pretty pronounced on cereals, but less so on field peas as a legume, and less so on sorghum that was in there, because the error bars were also bigger. I think there are two parts to the question. I would not really take the sorghum results very seriously, because they come from very few experiments, and that's why the error bars are so big. So there were many more independent experiments on rice and on wheat. On legumes, the idea is that legumes basically fix their own nitrogen through their symbiosis from air, and they can regulate that fixation. And so the original idea was that you won't see that decrease in legumes at all. I think the interesting point there is you actually do see it. Not so pronounced, but you do see it significantly. And Shania is working on legumes in our system, and she also, the first data, she clearly sees a decrease in nitrogen and even protein. So that is another probably argument that it's not a supply problem, because they could possibly fix more nitrogen. There's just some imbalance in the plant itself.
Natural vegetarians are screwed.
Well, I think, look, I'm not a nutritionist, so Luba said pretty much all vegetarians are screwed. I should paraphrase, should I? I didn't know.
That's your choice.
But the thing is, of course, that there is another paper by the same first author where he extrapolates these changes on different parts of the population. And the first thing is, of course, people in poorer countries will really significantly be affected because we already, in many countries, have a problem with, say, protein supply. And there's not only energy hunger in the world, there is also protein hunger. So you may have enough to eat in terms of energy, but you don't get enough protein. So yes, vegetarian is of course one thing, but it's not necessarily by choice in many cultures because it's just here it would be. But I think if you're well educated, you can change your nutrition to make up for it. But many people don't have that choice.
Oh yeah, 10:30. I've got one more question if there is one.
In your field studies, when you inject CO on your plants, the difference on CO, different heights, is it change or how you control it?
Okay, the question was whether the CO2 in the field is maintained across the different heights through the canopy. It's pretty good. We decided we don't need a second ring. Like there are systems, and the Japanese phase system in rice, actually they decided they have a second parallel ring, one just above the canopy, which is the one that we use. And the ring keeps following the canopy. And they decided they have a second one delivering CO2 a bit lower. The thing is, during the day, you have a CO2 gradient if it's reasonably not very windy. You have a bit of a CO2 gradient anyway, because you have very strong photosynthesizing canopy. And that's where we target it. It's the vertical, we know how much it is, but the vertical variation is not, we thought it's not worse than the natural vertical variation. So we disregarded it. But Mahabubur, our research engineer running the system, published a paper on these variations throughout the system. There is a recent idea to actually run it differently by basically not having the delivering ring, but having tubes hanging into the canopy and delivering CO2 at more points. It's currently in the development phase. I think they've tried it in Germany for a season. I don't quite know whether it's really that much of an advantage. The disadvantage is you have scaffoldings going in there to hang those things on. So you have more shading and you have other problems.
All right, well, we might call it a close there. Thank you very much, Michael, for